जिनिवाति दीनाम् अवित्रवतु।
I'll introduce myself to you. My name is Mrs. Shailaja Girishankar. I'm the head of the Department of Microbiology, Somaiya College of Science and Commerce. So, welcome to this session and lectures of genetics. Today we are going to do conjugation in bacteria, and conjugation is significant in that it's one of the process of transfer of genetic material from one bacterium to another bacterium. There are three processes, that is transformation, conjugation and transduction. And you can see that they are in a particular order, and it is in the order of discovery. That means transformation was discovered first, then conjugation, and then transduction. Now, all these mechanisms of transfer of the genetic material help in genetic recombination. Now, what's genetic recombination? Genetic recombination is when there is exchange of genes between two DNA to give you a new combination. It's an exchange of alleles between two DNA to give a new combination of alleles. So these mechanisms help in recombination. Why does recombination happen in nature? Maybe it is a way that there can be a lateral transfer of genes. Maybe it is to help the other members of the population to gain characteristics which can help in survival. Say for example, resistance to antibiotic. Bacteria are happy to pass on their ability to other bacteria so that they can also fight against antibiotic and they can survive. So possibly recombination, the purpose of recombination possibly is that. Today what we will be covering is the discovery, the X factor, the process of conjugation, what is an HFR, and how conjugation of bacterial chromosome, the passing of the bacterial chromosome by conjugation, can help in genetic mapping. Let's see when the conjugation was discovered. In 1946, two scientists, Vidyavihar and Prof, they discovered conjugation. What they did was they worked with two mutant strains of E. coli, and these mutant strains were for a particular growth factor requirement. So say for the first strain required meteorite and iodine for growth. The second one required leucine for growth. And when these mutants are grown separately on media, minimal media, a minimal media is the one which is not supplemented with any extra nutrients. So when these strains, two different strains, were grown independently on these media, none of them grew. Then what we did was they mixed the two strains, and then they take it on a medium, that is the minimal media, and they found that they were convinced that there was growth, which meant that they helped each other, these two strains helped each other to overcome their deficiencies, and therefore they could grow on a medium. So this led to the discovery of conjugation. Of course, they ruled out that no transformation happened. What is transformation? Transformation is the transfer or the transfer mechanism where a native nucleic acid, a DNA, gets into a recipient cell. Transduction, where the DNA is transferred from one bacterium to another bacterium through a fudge. And what they did was, they did an experiment to make sure that no transformation has happened. They also had a YouTube experiment where they separated the two strains, mutant strains, such that only the molecules which are synthesized can pass, because they thought that possibly the strain 1, which required biotin and methionine, could not synthesize, but the other strain must be making it and passing it on to the first strain. So they prepared a YouTube with a filter such that only those big molecules can pass through, and then they isolated the strains on minimal media gain from each arm of the tube. What they found, again, there was no growth. That means no biomolecules were passed, no nutrient molecules were passed from one arm to the other. So they found that it's not transformation, it is not just exchange of nutrient molecules between the two strains. They made sure that it was a transduction was not there, so they couldn't rule out that. So they found that there has to be a cell-to-cell contact, and of course it was also checked microscopically. That's how conjugation was discovered. What they found was, in 1952, you can see that William Case found that there is something called plasmid which was responsible for this, and he called it as a F factor, which is a fertility factor. What is a plasmid? A plasmid is a small, circular, extra chromosomal DNA that can give certain characteristics to the cell. And these plasmids are actually dispensable for the cell. The cell can do without plasmids. So there are types of plasmids. There are conjugative plasmids and non-conjugative or self-transmissible plasmids and non-transmissible plasmids, which means plasmids which can conjugate with another bacterium and transfer its own plasmid. These are transmissible plasmids. Non-transmissible plasmids are those which are present in the cell but cannot pass on to another cell. They can pass on with the help of a conjugative plasmid. Say, for example, called E1. It's a non-conjugative, it's a non-transmissible plasmid, but when it can hold hands with the transmissible plasmid will take it along with it and go into another cell. This is called as mobilization, which says that a transmissible plasmid can mobilize a non-transmissible plasmid. There are many plasmids which are transmissible, and an important one for your interest would be R factor. R factor is a plasmid which is responsible for many of the drug resistance. They are also transmissible, so you can imagine what happens when bacteria busy transferring those genes to others in the population, making them all resistant. Sometimes the plasmids can be passed on to other species too. So you can now think about the impending danger of this process, which is definitely an advantage to the bacteria, to the microorganisms, but a challenge for us. So F factor is that plasmid which helps in conjugation. It's a conjugative plasmid. So they called all those cells which has got F factor as F plus F. And all those bacteria which did not have a F plasmid, they called it as F minus. They also use the term maintenance. F factor being a fertility factor, they call those cells as donor cells. So these are donor cells. These are recipient cells and the donor cell will pass on its plasmid to the recipient cell. The ratio of an F plasmid to a chromosome normally is around 1 copy for every chromosome that is present. So what you can do is easily pass on the plasmid to an F minus and then make it into a F plus cell. So what does it mean? If you have F plus cross F minus, what you get? An F plus, which means you have a population in which there is F plus and F minus. Super. You can have all of them being F plus. All of them can be F plus. Does that make an F plus which has donated F minus? No. Because it has got another copy for itself. Therefore, it will always remain a F plus. Right? So, these are F plus cells. What about then the genes on these plasmid? There are significant parts to these particular plasmid of F factor. One will be ORI-TRA, ORI-C. There are TRA genes and some IS elements. These are the components of an F plasmid. TRA-O is the origin of transfer of the plasmid into the recipient cell. That's the point where a single strand break happens so that a single strand of the plasmid enters into the recipient cell. That's the phase. ORI-C is the origin of replication for plasmid. The plasmid replicates exactly like a bacterial chromosome. That is the rolling circle model. TRA genes, they consist of all those genes which are required for the actual conjugation process, for the production of the sex pili, transfer, connection to the recipient cell, actual transfer. All these, the proteins which are required for this are produced by the TRA genes. There are a few IS elements which are also present in the F plasmid. The actual process of conjugation. What happens exactly in conjugation process? There are male cells, there are female cells. The male cell, which is a donor, which is a F plus cell, has got a sex pilus. Sex pilus can be identified using male specific patches. So if you take an F plus cell and if you add some patches which are specific, you can find that these patches will go and get attached to the sex pilus. That's how you can identify whether the sex pilus is present or not. Corresponding to the recipient cell are receptor sites where the sex pilus can get attached. So if this is the cell and if this is the sex pilus, this is an F plus or an F minus, there will be a corresponding receptor site at which a contact can be made by the sex pilus. And once the contact is made, the conjugation bridge is formed, it gets contracted inside such that the cells come in direct contact with each other with a narrow bridge between them, through which the plasmid and plasmid can enter the recipient cell. How does the plasmid enter the recipient cell? They have come now very close to each other. This is your F minus cell, recipient cell. and you have, this is the bacterial chromosome present in both the cells. You have the plasmid. This is a double-stranded structure. So, of the double-stranded, one single strand will enter the recipient cell. And as it enters the recipient cell, things happen in the donor cell too. What happens in the donor cell? A new strand gets synthesized, or already it starts undergoing synthesis. As the F plasmid enters the recipient cell, it will circularize inside. It will circularize, and this also will undergo resynthesis, will undergo a replication. So what does it mean? At the end of the conjugation process, you have an intact F plasmid, one strand of which has entered the recipient cell, and inside the recipient cell, that has again undergo replication. So you have a double-stranded plasmid, F plasmid in the recipient cell, and you have the double-stranded F plasmid in the donor cell. So nothing lost, everything gained, right? And as I told you, the replication happens exactly like the replication in the bacterial chromosome. So this is the actual process of conjugation that happens. If you take a cultural F plus and cross it with F minus, this is what happens. Sometimes what happens is a recombination happens between the plasmid and the bacterial chromosome. Recombination is exchange of genes or alleles between two DNA. Here, the plasmids can get integrated into the bacterial chromosome, and when it gets integrated, you have a big structure of DNA. Part of it chromosome DNA, part of it is plasmids. So you have a circular chromosome, you have a plasmid. Do not look at the size. Okay, the hand can give the same size. So one, the chromosome, the other plasmid, and it integrates. You get a big circle. Now, when this happens, we call that cell as a high frequency recombination cell. HFR. So this is a F plus, and this is HFR. What is the frequency with which HFR is formed? It's 1 in 10 raised to 5. The frequency is 1 in 10 raised to 5, where a plasmid gets integrated into the bacterial chromosome. Then what happens? Conjugation can happen, because the plasmid genes are expressed, conjugation can happen, and at the point, that is the urethra, the plasmid can enter the recipient cell. It will enter the recipient cell, and it will carry a part of the bacterial chromosome along with it. If I were to number the plasmids and the bacterial chromosome, and if I were to draw this as the plasmid and this as the bacterial chromosome, let's put put some markers on them. Well, A, B, C, etc. They are markers. What are markers? Markers are segments of DNA or genes which help to identify the movement of the alleles across the population, individual, tissue, cells, or DNA. They help to identify the movement of alleles. They are normally the traits, so that you are easily able to identify phenotypically. I've marked this arrow at the tape here, just to show that this is where the leak can happen, and endonuclease normally makes a hit, and the origin of transfer will happen here. So the first few genes which will enter into a recipient cell will be those belonging to the plasmid first, followed by the genes on a bacterial chromosome. So how many bacterial markers can enter into a recipient cell along with the plasmid? Not many. Normally the order of transfer is around 50 kilobases per minute. That's your rate at which the transfer happens. And for an E. coli chromosome to enter into a recipient cell, it will take 100 minutes. This is a very, very rare event that could happen, that the entire E. coli chromosome can enter into a recipient cell mobilized by a F factor. This is because the natural shearing forces in the environment will not let the two cells be in conjugation for a very long time. So they come, they conjugate, transfer few of the markers, chromosomal markers, along with the plasmid genes, and enter into the recipient cell. So now what happens is, in the recipient cell, you have the chromosome which belongs to itself, and you have few genes of plasmid which has entered, and the part of bacterial chromosome which has entered. Can I then call, obviously a recipient cell is F minus, so when I have HFR cross F minus, can I say that I will get an F plus cell? Like F plus, when you cross with F minus, you got an F plus cell. So can I say HFR cross F minus will give you an F plus cell? Will it be an F plus cell? The HFR will not transfer all the plasmid genes into the F minus. So can I call it an F plus? No. So because to call it an F plus cell, it should have the entire plasmid, the F factor, the entire F factor in the cell. So in HFR cross F minus, the entire plasmid does not reach the recipient cell. So can I call it a complete F plus cell? No. So it cannot be an F plus cell. Okay? So an HFR cross F minus will not give me an F plus cell. Will not give me. Because the entire plasmid is not transferred. Once the bacterial chromosome enters the recipient cell, depending upon the homology between the markers which has entered and what is present on the endogenous chromosome, that is the chromosome which belongs to the recipient cell, depending upon the homology, recombination can happen. Two types of recombination are there: homologous recombination and site-specific recombination. So homologous recombination can happen depending upon the homology, and the way you can find out that is to have a recipient cell which is having a marker, which is having a trait different than the donor. Say for example, if your recipient cells are having, if these are A plus, B plus, C plus, have a recipient cell where it has got markers A minus, B minus, C minus. And if the recombination happens in that region, what you get a recombinant is A plus, B plus, and C plus. So that's how you find out. Then you can put it on the selective media which can select for the markers. And then you know that the markers have got exchanged. The stem cell has got these markers, the conjugation happens, and your experiment is successful. This process of conjugation, where a part of the bacterial chromosome is taken into the recipient cell, is a very good tool. It's a very good tool for genetic mapping. That is gene mapping. What is gene mapping? It is finding out or determining the location of the different genes on the chromosome in terms of the distance between the two genes or the loci, in terms of time period. So genetic mapping is one of the applications of conjugation. If you have an F plus culture, you can have HFR varied, which means the F plasmid can insert itself, can get integrated into the bacterial chromosome at different sites. So depending where it gets integrated, if this is the bacterial chromosome, maybe the F plasmid can get integrated here, can get integrated here. Different points. Depending on the number of points, I will get number of HFR strains. So for the same bacterial chromosome, for the same F plasmid, I can have different HFR strains. So using the different HFR strains, I can do the mapping. And this is because the order of entry of the markers into a recipient cell will depend upon, one, the site of attachment. As I said, it can attach anywhere. So the site of attachment, and also the polarity of attachment. What does that mean, polarity of attachment? When you have the plasmid like this, and the bacterial chromosome like this, they integrate by crossing over. So, and then you have a circle like this. That's because you have a particular polarity which integrated between ABC and XYZ. Now, what if this plasmid got attached this way? How will it look? One, two, three, 4, 5, 6. The same thing. I'm drawing it. But this time, how will it be? 6, 5, 4. This will be 1, 2, 3. Okay. So, oh, you just put it the other way. Now what happens? 1, 2, 3. Z, Y, X. So which are the last markers to enter? C, P, A, and 4, 5, 6. Okay? The polarity of attachment was different. Here I have changed it this way. Right? The polarity of attachment got different, and therefore the order of marker also differs. Now this we can consider when we are mapping the. There are cells of F prime, because when HFR, when it integrated with the dactylic chromosome, at a particular point of crossing over, if the second crossing over happens, it can dissociate itself, giving you back your F plasmid, entire F plasmid and your dactylic chromosome. But if the second crossing over happens at a site other than the first, then what you have is a plasmid coming out with a part of the bacterial chromosome inserted into it, and the bacterial chromosome losing a part of it. So you have an entire plasmid, 1, 2, 3, 4, 5, 6, which has got a part of the bacterial chromosome picked up from the chromosome, entire chromosome, and both are in the same cell. This now can act as a donor because it has got the entire plasmid which can get transmitted into a recipient cell. So the difference in transfer will be, in the first one when you cross an HFR and an F minus, you will not get an F plus, you will not get a donor cell. But if you have a F prime cell which can cross with F minus cell, and if the F prime has a FG node, which means the plasmid with a part of the dactylic chromosome in it, that can now act as a donor cell. It can transfer that into another recipient cell as an entire plasmid with a part of dactylic chromosome. So that recipient cell now can act as a donor cell. So this is conjugation in bacteria, where the two bacteria come together, cell to cell contact, and the transmissible plasmids, any plasmid which is able to transmit itself, can transfer into the recipient cell. They can help the non-transmissible plasmid by mobilizing them. And F factor is a fertility factor which can create conjugation and transfer even bacterial chromosome as an HFR cell. Thank you very much. I will just. So the conjugation machinery, as we saw, the donor chromosome, the recipient chromosome, the F villi, and the relaxosome complex, that is the relaxase enzyme and other proteins which are required. One is an endonuclease, the other protein, one endonuclease, which has to make a nick on the double-stranded F plasmid. This is a conjugation which happens the way it is shown in the diagram. Formation of the HFR strains. Two ways by which HFR strains can be formed in a culture: homologous recombination and biallelic confusion. The formation of the HFR strain. Interrupted mating experiments are those experiments which help in genetic mapping, where the HFR and F minus are allowed to mate, and at different time intervals, aliquots of the culture are taken and they are put on selective media to find out the frequency of transfer of a certain marker which is expressed phenotypically. The order and the timing of gene transfer can be used to determine the location of the various markers on the E. coli chromosome. This is how the graph will look after you've got the numbers of the selected organisms on selective media. You count them on the selective media, and then you can plot a graph which helps you to find out the highest frequency of the transfer. Obviously, the highest frequency means the first marker to get transferred, and in that decreasing order, the markers are farther away from the origin. Thank you. Any questions? Any questions? Can you think of more reasons why recombination must be happening? What would have been the implications? Is it the recombination which generated different species? Recombination is exchange of between two DNA, giving you a new combination of genes. So could you please that? What is the purpose in nature for recombination? Yeah? Something to ponder on? More than what we know? The whole idea is make your population stronger. Bacteria is being successful. Okay? Man finds out more ways, and they find equally smarter ways by passing on their ability. It's not only the antibiotics, it's the ability to degrade. There are degradative plasmids which can be passed through conjugation. So the bacteria says, I can't degrade many things, but my friend said, okay, take this plasmid and then you can cook your own food and eat. So they pass on some plasmids, degradative plasmids to me, and then I can now eat even pet food, plastic food, whatever it is. So aren't they helping each other?
Yes.
They are helping each other to make that a better community. And I think that is the greatest challenge for human being. When people get together for a positive thing, yeah, making it a better place to live, that's what bacteria is doing all the time. Okay? And possibly, we as scientists coming together and finding better ways to handle this, maybe that's how the science community will come together. We will also equip each other to do the same. Right?
Yeah.
So, wishing you all the best to reach the top and work as a community, you can produce great results. Thank you very much.